I'm Martha Diepenbrock, and I see lots of familiar names in our meeting tonight. I'm the executive director, fairly new at the Yellow Basin Foundation, and Corky, who usually hosts our Flyway Nights, has an event with her kids. So I'm stepping in as your host, and really excited tonight to have Mackenzie Miner with us to present what's happening with the Chinook salmon in the lower Poudre Creek. And some of you know there's a lot of current issues and things going on all the time in the dynamic area of our wetlands. So I don't want to take too much time except to thank you for coming. We've got people still coming in, and we'll continue to have them come in. So we'll wait a minute, I think, or two, Mackenzie, before getting you going. Mackenzie's organized her talk to stop at a couple of points, so there'll be time for questions along the way. And you can also type your questions into the chat, and that way we can take a look at those as she's going along and include you in the conversation. We plan to go for an hour, so we'll have time at the end also if Mackenzie finishes up, for those of you who want to stay on a little bit to answer any final questions that you might have at the end that may not have been addressed. So it's exciting. We're up to almost 50 people tonight. So I know there's a lot of interest in what you have to say, Mackenzie. So I'm excited to turn it over to you. I do want to add that Mackenzie is also one of our fellows, and maybe you could address that a little bit too, Mackenzie, but I will turn it over to you to begin your presentation, and thank you all for coming.
Okay, let's see about this. Okay, are you guys able to see my screen?
We are. I'm gonna then mute myself too.
Okay, and everybody can see it in presenter mode? Okay, awesome. Well, thanks everybody for being here tonight. I'm really excited to be here. Thank you, Martha, for the introduction. Yeah, Poudre Creek salmon and ecological research are some of my favorite things to talk about. And so tonight I'll be sharing with you some of the research myself and my colleagues have been doing to monitor and better understand the salmon in Poudre Creek. I'll try to frame for you a broad picture of salmon in Poudre Creek by discussing some of the ways we monitor salmon with an emphasis on our most recent data this last year. I'll provide a brief history to better highlight the larger impact Poudre Creek has on salmon reconciliation, and I will, as Martha mentioned, briefly address what we know about this year's salmon returns. Now, many of you probably know the history of Poudre Creek, but I'll start with some far back background nonetheless, so that we're all on the same page to appreciate the life this creek has had. The historical ecology of Poudre Creek is very different from the ecology which we observe today. Waterways would have meandered across the landscape through dense wetlands, as well as rivers, providing great diversity of habitats and organisms, salmon among them. This image is clearly an artist's rendering, but in her book, Forgotten Landscapes of California, Laura Cunningham provided this image of what I think a lot of the low elevation habitats characteristic of the Central Valley would have looked like 500 years ago. And it seems pretty obvious to me how much of this diverges from the creek we see today. Through the construction of levees, water diversions, and the urbanization of the surrounding landscape, the natural history of lower Poudre Creek and most rivers today has changed dramatically. So then if we skip forward a couple of centuries, we can see how much these landscape changes have affected salmon habitats. Let me get my pointer out. Okay. So here on the left is an image of what would have historically been accessible habitats for salmonids. And on the right in black, you can see how much habitat is no longer accessible. And I think this study was conducted specifically with respect to steelhead, but it still clearly illustrates the presence and absence of anadromous habitats. But by rehabilitating, reopening, and reconciling habitats like Poudre Creek, we can begin to understand how to shift this imbalance of available habitat. And again, most of you probably know the more modern history of the creek, but through various mechanisms and droughts, like many other systems, salmon were eventually extirpated from Poudre Creek when there wasn't sufficient flow or habitat to support them. And here are just a few images of a dewatered creek. And I think this picture on the top left was actually taken around Mace Boulevard and of course, Monticello Dam. However, thankfully, a group of dedicated and persistent citizens pushed for more water in the creek and worked with the water agency through the legal system in the 1990s to sign what is now the Poudre Creek Accord in 2000. This dictated the use of a natural or functional flow regime, as I'll refer to it, to create seasonal pulse flows for lower Poudre Creek and to assess annually if and how native fish were responding to the new flow regime. But often I think it's important to frame, what does it mean for a creek to have a functional flow regime? And well, in our situation, it means that the constituent parts of a hydrograph facilitate ecological function. And since this natural or functional flow regime is kind of serving as the backbone for returning salmon, I'd like to address that really quickly. The Accord addresses in part flow requirements for lower Poudre Creek, prescribing not only minimum water flows to keep the creek wetted, but also strategic flow patterns. The pattern of flow is designed to mimic how the creek would have worked prior to dam construction or regulation. And so here in this picture, the blue line indicates what the hydrograph of a happy, healthy creek might look like, or a creek that's unregulated. And as time goes on the x-axis, the height of the line on the y-axis indicates how much water is in the creek at any given time. You can see various peaks and valleys in this blue line. And these would have been periods of high runoff from rain events or melting snow in some cases. And so fish that evolved to live in these oscillating environments developed strategies to help them survive, like laying eggs when flow and temperature were adequate for their offspring, or timing migrations when there was enough water to help push them along. However, when a dam is constructed and flow in the creek becomes regulated, a lot of times these peaks and valleys can disappear, and suddenly the life history strategies these fish developed don't make sense anymore. And so this is when the concept of a functional flow regime, or sometimes a natural flow regime, comes into play. So if we can identify which flow patterns are most important, water releases, in our case from the Poudre Diversion Dam, can be timed to mimic them. And this is demonstrated in the red line here. So we can see a smaller pulse flow in the fall, possibly as a tool to attract up-migrating salmon. And then second, a peak flow in the winter that might be important for shaping the riverbed or performing geomorphic restoration. And finally, a late spring pulse, possibly to help juvenile salmon out-migrate. And here you can see the type of natural or functional flow regime that has been formulated and implemented in lower Poudre Creek. So what I have pictured here is what the hydrograph in lower Poudre Creek looks like today. This is from the most recent water year. And here you can see again the wet season initiation flow, which oftentimes we call and we use as like the salmon attraction flow. And then a peak flow in the spring that we often try to time with juvenile outmigration. So these fluctuations in a flow regime and the environmental variability are critical to salmon management because they rely on cues to know when it's time to move in or out of freshwater and saltwater, and then it helps them to do so. So the lives of salmon are truly characterized by change, and to me, that's reason enough to find them inspiring because I think change is really hard. But when we talk about salmon, why is Poudre Creek relevant and important when we tell the story of salmon? And it's because nearly half of all of California's native salmon and trout run the risk of extinction in the next few decades. And these days, I think it can be really hard to find encouraging tales of fish. However, the return of salmon to Poudre Creek is encouraging and it has inspired action. And while fish may be in hot water, as we continue to navigate droughts and warming temperatures, I think that Poudre Creek can serve as a refuge for salmon and may provide us with an opportunity to study reconciliation for other subpopulations. This image I have here is from a report compiled by the UC Davis Center for Watershed Sciences, as well as CalTrout, and it kind of details the status of salmon in California and trout. And so if you're interested in learning more, I've included a link here that will take you to the report. Okay, so now I'm going to walk through some of the data we've collected in the last year and how we account for the various life stages of salmon in Poudre Creek and what that can tell us about recolonization. Since I'm focusing on recent data, I'll start with the juvenile population that we monitored this last spring and then work my way through time to this year's current adult returns. And I'll note really quickly that since I'm focusing tonight on more recent data, if you're interested in learning more about the last several years of salmon data, our lab group has given a couple of other presentations, as well as Peter Moyle and Rich Marovich. They've also given some really enriching talks that are available on YouTube. I think if you search like Poudre Creek salmon, those talks will come up, and those detail a little bit more of the previous salmon data. But anyhow, understanding the dynamics of the juvenile population is important because while rates of returning adults are critical to establishing a salmon run, survival by the juveniles is how we maintain a salmon run. You can't have a population without meeting all of the life stages. So to assess the fry and smolt stages, we capture them as they move downstream in downstream migrant traps. And one example of a downstream migrant trap is a rotary screw trap, which is pictured here. Monitoring them in this way tells us, number one, if the adults were successful in spawning. It also gives us an idea about production in the creek, and it begins to build a picture about the timing of their movements and the conditions in the creek. And during this time when we're trapping fish in the spring, this is also when we perform acoustic telemetry studies that allow us to track the relative success of salmon outmigration. And so I'll explain really briefly kind of what a rotary screw trap is, because I think it can seem a little bit daunting, right? We put the giant metal thing in the creek and catch fish with it. But when we use a rotary screw trap, we try to place it in a section of the creek where a majority of the flow will flow through this big metal drum right here. And so as the flow meets that cone, there's baffles in the center, and there will be a picture to show you those baffles. But as it hits the baffles, the cone spins, and then the fish are pushed into this box that's located in the back right here. And so then we go out every day, sometimes multiple times a day, to check that trap. And so in this way, the trap isn't picky about what it's fishing for. So we capture salmon as well as a lot of other native and non-native fishes. And so as we capture fry and smolts, we always try to collect as much data from these fish as we can. So we'll collect size data, genetic data, and timing data. This slide shows some of the most recent trapping data from this last spring in 2021, as well as another image of our rotary screw trap from a different perspective that is maybe a little bit of a better picture to kind of show you those baffles. This top graph illustrates our capture rates as it relates to creek discharge. And discharge in a creek is a measurement of volume. It's measured in cubic feet per second. So it's the amount of water moving down the creek at any given time. And the values for discharge that I'll report to you are in mean daily discharge. But here you can see what I think is a slightly delayed, if I can get my pointer in the right spot, is a slightly delayed response to the spring pulse flow. You can see a peak in the pulse flow, and then all of a sudden, all these salmon are coming downstream. And then after that, you can see fairly consistent capture rates for the remainder of the season until the tail end of the outmigration window. And then this bottom graph reflects water conditions during our trapping window. This yellow-orange line right here is the daily mean water temperature reported in degrees Celsius, which is reported on the left axis right here. And then the blue line is dissolved oxygen reported on the left axis. Again, this is mean daily concentrations. It's worth noting really quickly that temperature and DO are inversely related. So colder water holds more oxygen. So it makes sense that as water temperature begins to rise at the very early part of summer that the DO starts to decrease a little bit. And so these capture rates, as we find them in the spring, are variable between years in their timing. Sometimes we capture more salmon in March, and sometimes we capture more often in May. Because of the timing of juvenile outmigration depends on when adults are able to. So this whole shift back to that year's timing. The run in Poudre Creek is a fall run salmon population, but because the timing of the adults moving up into the system is really variable, it can be biased late into the season, kind of following more of a late fall run timing. But you can see here that the bulk of the juveniles have commenced out migration before the temperature gets too warm. So after we capture the juveniles in a screw trap, hopefully ocean, and this transition from a river or a creek to the nutrient nutrient-rich habitats of the ocean is considered one of the most vulnerable periods in a salmon's life. So we do our best to capture that transition and understand what is influencing survival. But as John Shepard said, managing fisheries is hard. It's like managing a forest in which the trees are invisible and they keep moving. So after a few seasons of conducting acoustic telemetry studies, I can tell you that it's a little bit tricky. But to do this, we tag fish with a small transmitter that has a unique identifier. And then we deploy many receivers, kind of like this one, throughout the creek. And so biotelemetry, as we use it, is the remote measurement of behavioral data. And so after we tag a fish with a transmitter, which is kind of pictured in this top picture, this is the transmitter right here. That transmitter will communicate with the receiver. And then that receiver stores the date, time, and location that that fish passed it. And then that's all stored on the receiver until we go out and get it. And so we use active telemetry, which means we mark one fish one time, and then we recapture that fish on a receiver. And so this is kind of neat because we don't have to continually interrupt their movement patterns to get an idea of where they are, how long it's taking them to go a certain distance. And so pictured here is one of our tagging events from this last season. As you can see, it kind of takes a crew of people to do this. We have somebody tagging fish here, somebody recording all the metadata, and then people processing all the other fish that we capture in the trap at the same time. And so you can see here where those receivers were deployed throughout Lower Poudre Creek this year. This year, I designed the array in the hopes of identifying and assessing possible passage barriers and to begin to characterize juvenile salmon outmigration in Lower Poudre Creek. The creek was segmented into about 7 reaches using acoustic receivers located at the top and bottom of each reach. Unfortunately, all these reaches are of slightly different length because it can be tricky to get into the location at the creek where we want, when we want, and it's the right condition for putting a receiver. So there's a little bit of, section A is a lot longer than section E, but passage barriers were identified. One of them was an agricultural impoundment dam, which a lot of people call the check dam. The other passage barrier was an unusually large beaver dam, and the third was a road culvert.
Mackenzie, there's just a couple quick questions in the chat. One is, what species of salmon are you studying? And then the second one is, how many fish are recaptured on a receiver?
Cool. So I'm sorry, these are fall run Chinook salmon. And the number of fish recaptured on a receiver varies depending on how many fish make it to that location. But this year, out of the screw trap, we tagged 170 juvenile salmon with those transmitters. And then as they move down the creek, you'll be able to see, I have a graph that will hopefully illustrate that, just how many fish make it to each receiver. So really quickly, I will mention that while the data I'm sharing with you tonight is lower Poudre Creek specific, there is a larger array of receivers throughout the rest of the Sacramento River and in the Delta that's maintained by a collaborative network of researchers. And so our salmon could get picked up on those receivers. So hopefully in the future, as that data becomes available, we can extend our questions and some of the data that I'm going to share with you tonight to include that. Okay, so possible passage barriers. Of the possible passage barriers we identified, the first one that fish would encounter as they're moving downstream would be this really large beaver dam. And so we actually discovered this beaver dam during our last carcass season in 2020. After we stopped observing salmon upstream, we went looking for them, and this is what we found. It turns out that earlier in the fall, when the water levels were higher, adults were able to pass over this dam, but then later, as water levels receded, it became impassable. So this year, I wanted to know if this was something that juvenile salmon could breach or if it impeded any outmigration. I don't know if this picture is a great illustration of it. It is a 2.5 meter tall beaver dam. And in the foreground here is me in a boat. And I am like not even halfway up this beaver dam. And unfortunately, actually, for the next passage barrier, I don't have a picture of a road culvert, which is really silly. I need to remember to take more pictures. But if you've seen a road culvert, you've seen this road culvert. But then the third passage barrier that we identified was this agricultural impoundment dam, or the check dam, pictured here on the left. So these flashboards come out seasonally. So it's like a semi-permanent dam. But when they're in place, I think it's pretty unlikely that juveniles are able to successfully navigate this. This is the only way in and out of Lower Poudre Creek. There's no other way around or through. They have to go over, essentially. And so, as you can see here, there's not a lot of directionality. A lot of the flow downstream of this check dam is actually coming from the seepage that comes between these boards, right? So behind these boards, a really large pool forms. And so salmon get to this pool and have to figure out where to go from there. And then on the right is a picture of what Poudre Creek looks like downstream of that check dam from the vantage point of standing on top of the dam. And so you can see the water's a little bit slower. It's pretty channelized and pretty heavily vegetated. Okay, so now I'll share with you some of the results from the telemetry study that we did. And this is where I said, hopefully it illustrates to you how many fish make it to each receiver. So this histogram plot shows detection histories from this year. And these river kilometers might look a little wonky because we know that Poudre Creek is not 175 river kilometers, right? But that's because we measure from 0 river kilometers at the beginning of the delta at the Golden Gate Bridge. And so what this is saying is that from our release site at the Rotary Screw Trap, juvenile salmon have about 176 river kilometers to go before they make it to the ocean. And each gray bar is essentially the number of fish detected at each river reach, so at that river kilometer. And then on the x-axis is the number of unique transmitters detected, because each transmitter has a unique ID. That ID belongs just to that one fish. So at the top is the release site, which is downstream of our screw trap. And then you can see how as the salmon move downstream, the areas that might be more hazardous. So where the greatest change in number of fish counts occurs. So looking specifically at those passage barriers we identified, it appears that the beaver dam, kind of highlighted here in blue, this top bar is the receiver located right above the beaver dam, and then the receiver located immediately downstream of the beaver dam. Doesn't appear to have a huge impact on outmigration, and neither does the road culvert, right? This bar right here, this 24.1% is right above the culvert, and then 18.2 right below the culvert. But there are a few areas that we do see have an impact on the number of fish detected between those reaches. One of them being the uppermost reach, so the transition between the receiver located downstream of the release site to about Russell Ranch, which is located downstream of Winters, but upstream of Davis, if that helps orient you. The transition between this receiver and this receiver is about 6 river kilometers, so that may account for some of this change, right? Like at these lower reaches, there's a little bit less change in distance, and this one is a little greater. Another one that stands out is this check dam, right? The distance between this upstream check dam receiver and the downstream check dam receiver is not very much. So it appears that a lot of fish made it to that big pool above the check dam and then either got confused and couldn't figure out how to navigate that pool and make it downstream before they, met their fate some other way. And it's worth noting that this is just a qualitative look at these detection histories. I haven't yet run any rigorous statistical tests to tell us which reaches significantly influence survival, but this data can be helpful nonetheless in deciding which areas warrant more investigation now. So this plot, again, looking at the telemetry data, looks a little closer at the in-reach behavior of fish. And this is intended to illustrate the average time spent in minutes in each reach by the juveniles. So we can see that the residency time was greatest in the upstream reaches, where habitat quality is likely better compared to the lower reaches. So fish may be holding in the upper reaches of the creek, but seem to display a slightly more consistent migration pattern through the downstream reaches. In addition, they seem to be spending a little bit more time at that very last reach that's just above the check dam. Again, it may be possible that they're getting to that pool behind the check dam and spending some time there trying to figure out how to navigate it before meeting their fate or passing over the check dam. So ultimately, what all this telemetry data tells us is where fish are spending more time or what areas may be acting as mortality sinks, but more research and data collection is needed to be performed in those areas to determine why that might be. Okay, so now that we covered the juveniles, I will transition to talking about some of the adult data. But before I do, I can answer some questions in the chat or any questions that I missed. I know the telemetry data can be a little hairy.
So, Mackenzie, let's see, we talked, so how will this project help fisheries managers manage fall Wren Chinook salmon as a whole, including the mainstream?
This here, I can bring the chat up, sorry. So specifically the telemetry data and the screw trapping data, I can think of a few examples. So just this last year, as I mentioned, the timing of the juvenile outmigration can be very variable during the year, depending on when the adults were able to spawn. So if the adults are able to spawn earlier in the year, we'll see juveniles outmigrating earlier. And then if they spawn later, the juveniles will outmigrate later. And so that will largely impact when it's most beneficial to release a pulse flow for salmon. You don't want to release a pulse flow before all the juvenile salmon are ready to outmigrate or too late, right? You really want to we have a relatively little amount of water to work with. So we really want to optimize when we time those pulse flows for salmon, if that's how we're going to use the pulse flows. In addition, the telemetry data, I think, could be used to identify which areas of the creek are most hazardous or are most in need of restoration. There are certain sections of the creek that we can walk by and say, oh, that's bass habitat, or that's pike minnow habitat or something. And so if we can identify those areas and that's reflected in the telemetry data, maybe we can start to target our restoration efforts to those areas that are most hazardous for salmon.
Let's go down the next few questions in the chat. We are down just past user. What is the total number of transceivers installed in fish?
So the number of transmitters that we put in fish varies year to year. It kind of depends on how many fish we're capturing. Some years we're not able to capture as many juvenile salmon as we would like, either because the flow is too high and we can't safely operate our trap, or there weren't a lot of juveniles. Like in 2019, there weren't a lot of juveniles. I think we found a total of 28 juvenile salmon, and that's not necessarily a reflection of production in the creek, and is often just a reflection of when we're able to sample. But this year, we put 170 transmitters in fish. Generally, we try to budget to put at least 100 transmitters out there. And this year we got lucky. We found some extra transmitters. And, you know, from my perspective, if we're permitted to do so, the more fish that we can tag, the better, because I think it's probably more indicative of what's happening with a larger population. But we are limited because, as you saw, I hope in those previous slides, like it's a fairly large tag. You know, it's smaller than my fingernail, but when you're only this big, like that's pretty big. So we can only tag salmon after they reach a certain size. And so in that way, you know, you're already kind of biasing your telemetry results because you're only targeting fish that are of a certain size at that time of year. You know, it's totally possible that juvenile salmon are out migrating at an earlier time, and we're just not able to capture that with telemetry data because of technological limitations. I would agree, Maureen. The check dam could definitely use some passage upgrades. That would be fantastic. Who controls the check dam? And is there any coordination to accommodate the passage of salmon? I couldn't tell you for sure who coordinates the check dam. I think it might be operated by a private farm more often than not, but they have been really willing to coordinate with us and the water agency, and there is a lot of coordination to accommodate the passage of salmon. Rich Marovich, who I think is just a fantastic human, has done a lot to really coordinate salmon in the creek. You know, it's hard because despite having, you know, 6 years of data, there's still a lot we don't know. So we're kind of trying to like adapt on the fly. And Rich was really critical in this last year in trying to time the pulse flow, the spring pulse flow, such that it would benefit the most amount of salmon. And so our group at UC Davis coordinated with him to look at previous years' data to try to figure out when would most salmon be leaving the system and when would the pulse flow most benefit them. So these acoustic transmitters that we use, I haven't recovered any in adult fish, but their battery, I'm just gonna go back. I think I can do that. Oh goodness, this is becoming, okay. So this is the transmitter and this little silver part is the battery. And so the battery on those transmitters is only good for about 100 days. So even if they were coming back in adult salmon, it would be pretty hard to measure that unless we like did a really thorough dissection. But you know, people have Floy tagged and pit tagged salmon. I found my first Floy tag on a salmon a couple weeks ago. The mortality rate with acoustic tagging, at this point, it's not very high. Like this picture right here kind of demonstrates all the equipment we take out with us to do everything we can to mitigate any loss during surgery. And the group I work with at UC Davis has been acoustically tagging salmon for 6 or 7 years, I want to say now. So they kind of have it dialed in. And so the mortality rate I observe when I'm doing surgeries is pretty low. And then after the surgery, after I complete the surgery here and put them in a recovery bucket, I don't release them out into the creek to figure it out, I'll actually hold them for a period of time to make sure that they recover and that they're recovering in a, I don't know, I would call it like a less stressful environment. We have a cage that we put in the creek, and that where they'll all hang out for a day or so before I release them to make sure that they've recovered from any effects of the surgery or any of the dosing effects.
Mackenzie, Robin responded to the who operates the check dam. So you can see in the chat, Robin's response to everyone that it's operated by the Los Rios Farms as kind of part of a whole collaborative effort. So there's coordination, but as she said, it's complex and a lot of moving parts.
Yeah, I would agree. There are a lot of moving parts. And one of the things to consider when we're thinking about management in Poudre Creek is that it's not managed just for salmon. Poudre Creek is a multi-use resource that is meant to benefit, waterfowl and agriculture and urban use, so it's a kind of about finding a resolution that is beneficial to the most parties, right?
So Mackenzie, maybe you could jump back into your presentation and then we'll stop again near the end, because I know you've got more.
I do. Okay. I'll transition to adults now. And carcass surveys have been conducted annually by our group since 2016, but salmon have been observed returning to the creek pretty consistently since 2014, with a few observations of spawning salmon dating back to 2008. And so when salmon are present in the creek, we float it weekly from top to bottom, looking for carcasses. And from each carcass, we try to gather as much data as possible before marking that carcass with a jaw tag and putting it back in the creek. I'll just talk about a few things that a carcass can tell us about the life of a salmon, like where it's from, where it's been, and how old it is. As this little chart demonstrates, the number of salmon returning to Poudre Creek varies notably between years, and there can be several reasons for this. Sometimes it has to do with when the fish can access Poudre Creek, like when boards come out at the check dam or when the pulse flow occurs. And sometimes, like in 2019, fish weren't able to access Poudre Creek until much later in November. And by that time, a large part of the salmon run has finished. Or in years like 2017 or 2018, these returning adults are likely strays from other systems or hatcheries. That as juveniles in 2012, 2013, 2014, during the big drought, got trucked to the Delta. And so when fish are trucked through the Delta, they're unable to experience the river corridor as they normally would during outmigration. And so this results in something called non-sequential imprinting, which essentially means that as adults, they don't know how to sniff their way back to their natal habitats, which can lead to greater rates of straying to other habitats like Poudre Creek. I think sometimes we think of straying as a bad thing. Hatchery managers want their fish to return to their hatcheries, but in the case of Poudre Creek, straying fish is one of the only ways we're going to establish a new population. So in a way, it's kind of like as juveniles, they lay breadcrumbs that they can follow on their way back. And so this is a picture, I know it's a little morbid, of a few of the carcasses I think we recovered in 2017. But there are a couple ways we can try to evaluate the origin of salmon in Poudre Creek. And this may help us to understand why return rates vary so much. We can use coded wire tags as well as otoliths to do this. So as we come across carcasses during our surveys, we can guess they originally came from a hatchery if they're missing an adipose fin. It's pretty standard practice in California hatcheries for juveniles leaving the hatchery to be adipose clipped and coded wire tagged. And so if we come across a fish that is missing an adipose fin, we'll collect the head and send it to a lab at CDFW, where they can verify if it has a coded wire tag, if it's of hatchery origin, how old it is, and then if it was released off-site, where it was released. And a coded wire tag is pictured here on the left. It's a tiny, tiny embedded wire with a serial number on it here, and that serial number corresponds to metadata. So for those fish that may have been trucked to the Delta during the drought, this is a good tool to use to identify them. In addition to coded wire tags, we also collect otoliths from every carcass we recover. Otoliths are the inner ear bones of fishes. So from these otoliths, we can get a lot of information about fish growth, age, habitat use, and life history. Otoliths allow us to peer into a fish's history by using microchemistry and isotope ratios at different rings on an otolith. Like a tree, an otolith grows every year, right? And at that point in its growth, it lays down a ring, and that ring has an isotopic signature of whatever water it was in at the time. And so my colleague, Malta Willems, did just that for the otoliths that we collected in 2016. And again, I have a rather morbid picture for you of how we extract otoliths in the field. They are located in the head of a fish, and they're these teeny tiny little, they look like angel wings to me, right? Kind of, I think they're so pretty. But anyway, like I said, Malta analyzed the otolith microchemistry from those otoliths collected in 2016. This plot from his publication highlights the distribution of natal origins, and natal origins being where those baby salmon spent time in fresh water, and at least 7 origins were identified. And we can see that a great majority, 88%, were found to be from Central Valley hatcheries. However, there was information that pointed to a few wild fish in the system early in the process of recolonization. Remember, this is 2016, so this is pretty early on when we tell the story of Poudre Creek salmon. So in the future, we expect these numbers to begin shifting to the right as we hopefully see more fish of Poudre Creek origin return. Also from this data set, Malta was able to determine the age structure of the population. Most salmon returning to Poudre Creek in that year were 2 to 3 years old, mostly strays, again, from Central Valley hatcheries. It does indicate that salmon are able to quickly capitalize on restored habitats. And there is a plan to continue this work. Actually, we were talking about it this week, to continue to analyze everything, the otoliths from 2017 to 2021. And it was really fortunate that we were able to recover adults this year, because it's possible that, you know, through the use of otoliths and genetic samples, we'll be able to tie this year's adults with juveniles that left the system in 2018. It's not pictured here, but 2018 was a really big year for juveniles in Poudre Creek. So it's possible that now those juveniles that left in 2018 have had time to grow in the ocean and come back. And so, you know, we'll have to see what the data says. Okay, so this year, as I think many of you probably already know, there has been a distinct lack of adults present in the upstream reaches of the creek where we would normally expect to see them spawning. That's where a majority of the restoration activity has occurred, and there's suitable spawning habitat. So far, all of the carcasses we've recovered this year have been found in the lowest reaches of the creek, all within about 2.5 river kilometers of its endpoint. And the endpoint of Lower Poudre Creek is in the tow drain, where it meets the tow drain, and then the tow drain connects to the Sacramento River, to the Delta, and to the ocean. And so many of the salmon that we've recovered so far this year perished before spawning, as indicated by the many ripe females that we've recovered so far this year. In addition to their location at the bottom of the creek, this was kind of a clue to us that something was amiss, because most of the time salmon will at least attempt to spawn, even if it's in less desirable habitat. And so right now we believe that this premature mortality event is the result of really low dissolved oxygen concentrations. Do you guys remember that really crazy monsoon-like storm that happened at the end of October. This graph kind of illustrates that precipitation event, and you can clearly see here the event that I'm talking about. Right around October 24th, we got almost 4 inches of rain in less than 3 days. And here are a few plots that I think kind of demonstrate why we think it's low dissolved oxygen. This is all data collected from water gauging stations in the tow drain near Cortland, California. So that's downstream in the tow drain of where Poudre Creek meets it. This data is publicly available on the USGS website, and I've included the link to that website if you're interested in looking at more. They have a lot of other variables that you could look at that aren't dissolved oxygen or organic matter. But I'm trying to give you guys a rough idea of the factors that we think are involved. And again, this is all preliminary information. A lot of people are still actively looking into what happened. You know, nothing's definitive yet. These are just the data that I have to go off of. So on the left is dissolved oxygen, which is measured in milligrams per liter. And on the right is dissolved organic matter. So what I think has happened here is that all of the materials on the landscape, whatever that may be, you know, if it's plant matter or if it's waste, what have you, that has been building up over the last year. Because, you know, if we are reflecting on this last year, there hasn't been a ton of rain. And then all of a sudden, this crazy rain event, I think, mobilized all this buildup during the rainstorm and drained it to our rivers. And so that drastically increased the amount of organic matter in the water, which would then lead to greater rates of respiration. And, you know, then the bacteria would consume more oxygen. So you can see here a sharp increase in organic matter in the water, and then following that, a sharp drop in dissolved oxygen concentrations. And so all of this has essentially led to one of the lowest dissolved oxygen concentrations observed in the last handful of years. And actually, my supervisor, Dennis Cockrell, has put a lot of this information together, and he went back through the last 9 years worth of dissolved oxygen data from this gauging station and identified this blip here as the lowest point of dissolved oxygen for an extended period of time in the last 9 years. And so generally, dissolved oxygen readings of about 6 or 7 milligrams would be considered normal, but readings below like 3.5 or 4 milligrams per liter could become lethal for salmon, not to mention other fish, right? And a lot of other fish besides salmon were noted to have perished during this, you know, low DO event. And salmon are pretty hardy compared to a lot of other fish. So this data not only indicates, you know, possible causative agents of this premature mortality event, but also indicates that it's not just Poudre Creek that's experiencing this. The whole of the tow drain has pretty low dissolved oxygen concentrations. And I know that the Department of Water Resources monitors fish in the tow drain, and they have noticed fish mortalities as well. So, you know, I think what we can do is be aware of this for the future, which I think is exciting. The future of Poudre Creek is not set, right? And so we have the ability to make change here. And we're adding new components to the project every year as we learn more about Poudre Creek, right? It's all based on what we're learning now in adaptive management. And so I've been really fortunate to work with a lot of different people on Poudre Creek, and I'm really glad to be a part of, you know, the recolonization effort. And I think with that, I can answer any more questions that I may have missed earlier.
That's fantastic presentation and a lot of information. Really appreciate your work and, you know, the way you're talking and sharing, you know, what these issues are about and what's being looked at, Mackenzie. So there's a few other questions. It's kind of a big picture question about what are some resources, regulation, and management tools we can implement to support the longevity of the salmon run in Poudre Creek as global warming is shifting the natural world. Do you have any thoughts about kind of the big picture, things that are happening at the policy and management level, or things that, your team and your colleagues are working on in that way?
Yeah, I don't have a good beat on the policy stuff at the moment, but I do think that when we're thinking about managing salmon in Poudre Creek, I think kind of spreading awareness and knowing that it's a resource that we all share and that it's something that we're trying to protect for the future is important. I have a Poudre Creek salmon shirt that has Winter Salmon Festival on the back, and I'll be in the grocery store and people ask me questions about it. And, despite what I feel like is a lot of a Herculean effort in managing these salmon, a lot of people still don't know about it. And that's not to say that they don't care. It's just, they haven't been exposed to it. Additionally, I think as far as management tools go, the fact that we have, essentially a reservoir of pretty cool water, at our disposal is a really unique asset. We have to manage our water really carefully, but I think we can do it strategically. We have this water, and I think salmon will do most of the work for us if we can find a way to benefit them and use that water strategically to do so. I'm not sure that's super insightful, but we can circle back.
Yes, there's let's see. So
could a fish ladder be constructed around the check dam? There has been a lot of talk about, installing a check dam. And I know, folks at the Water Agency are more than willing to consider this and have worked with us on trying to find solutions. I think that there's been a lot of talk about a bypass channel around the check dam. And so rather than building a temporary fish ladder, the problem with the fish ladder is that we need to be permitted to do that. We need to be permitted to do anything. But if we build a bypass channel that finds the low spill point of the creek upstream of the check dam, then the check dam doesn't have to be so closely regulated for the upstream and downstream movements of fish. They could move freely. I don't know what the final status is on constructing a bypass channel, but I know that there are people working on it.
And then around Lake Solano Dam.
I don't know about Lake Solano Dam. When we're thinking about dams, the Poudre Diversion Dam and the check dam are not, very large dams. When we think about the fact that we have fish ladders up the Columbia River, at Lower Granite Dam and Bonneville Dam. I think, I've heard a lot of people talk about finding passage for fish around the Poudre Diversion Dam and getting them into that inner dam reach, which is really probably a good example of salmon habitat. But, I think that it would really benefit salmon to first kind of solve problems in this lower Poudre Creek, where we're seeing mortality. Because if, they can bust through, chunks of bad habitat. But I think if we can make sections of the creek more inviting and more hospitable for spawning, we could do that. But yeah, I don't think it would be infeasible to put a bypass channel or a dam around the Poudre Diversion Dam. Do you know what caused the low DO? I can't tell you exactly what caused the low DO. I can tell you what I think likely caused the low DO, and I think that it is all the stuff that had been sitting on the landscape for the last year or so. This is just what I'm inferring. If you talk to a water quality analyst, they may be able to tell you that it was ammonia. I don't know that. It could be any number of things. But whatever it is, I think it was introduced to the system after that really large rain event. There was a spike in organic matter, which may have increased anaerobic restoration, which consumes a lot of oxygen and depleted the oxygen available for fish.
Let's do it just a couple more. There's, Robin is responding to the design environmental analysis of that bypass channel, saying there was the problem that we ran into is the petroleum products pipeline that would need to be relocated. So it sounds like that's what people are still working on. Yeah. Oh, did we miss the toxicological analysis performed on the carcasses to see if some chemicals might have been ingested?
I have thought about that, and we have talked to our vet on staff. And the problem with finding a suitable carcass for like toxicology analysis is that it has to be a more bunned carcass, meaning that it has to be relatively fresh. And I don't know if those pictures that I had in the presentation were indicative or not, but by the time we get to the carcasses, they're not always very fresh. So we haven't yet found a carcass that's fresh enough such that the tissues are in good enough shape for that. There are people who are doing a lot of water quality testing. I know there was somebody out at the check dam a couple of weeks ago who's from, I want to say it was like the California Department of Environmental Investigations. They were there from like the Office of Oil Spills or looking for petroleum in the water. So there are a lot of people doing a lot of things, and I don't have that information yet, but hopefully it will all come to light.
All right. Okay, well, I think we're just probably a few more questions that will wrap up, and then maybe you could stay on a little bit longer, Mackenzie, if there are people that want to stay on for a few more. I just want to thank all of you for attending this, and again, thank Mackenzie for her work and her time to present this to all of us. We really appreciate it. We have, I'll just say our next talk is in a month. And we've got Joe Hobbs, who's the area manager for the wildlife area, who will be speaking next month. So some of the questions that you're asking kind of flow naturally to Joe, who's responsible for the management of the Yellow Bypass wildlife area. So we hope that you can join us next month in January to hear what Joe has to say about some of the projects going on right now, and some of the other issues that are all at play in managing this incredible multi-benefit open space, a resource that that is beloved to all of us. So thank you for coming. And if you want to stay on for a few more minutes, we will keep it open. Right, so why don't we just go ahead for those that want to stay on, Mackenzie, and see if there's any other questions that you can address. I think we were at Max saying that the low DO at the tow drain is a barrier like a dam. Max sounds like he's in the know. Let's see, I read the check dam was blocked and not opened up during the adult salmon run last fall. Do you know if that's true? Oh, Robin said not true. So Robin keeps track of all these things, even though she's not full-time with the Yellow Basin Foundation. Robin is still involved in all of these issues. So it's really valuable to have you on, Robin. All right, let's see. I think most people are just really thanking you for your presentation, Mackenzie.
Well, thank you for having me.
All right. So everybody have the rest of a good evening and a good holiday times coming up. Stay safe and keep track of the Yellow Basin Foundation and go to our website for all the other things going on in the wildlife area and other talks and tours. So thank you all.